Hi everybody, my name is Bridget if we haven't met before, and today I'm going to help us wrap up the semester continuing our discussion of phylum Chordata. Today we're going to be talking about frogs. So our objectives are being able to read and interpret cladograms, understand major distinctions between protostomes and deuterostomes, and explain how frogs perform basic life-sustaining functions, so how they're filling their ecological niche. So let's start off with a background on cladograms, protostomes and deuterostomes, and chordate characteristics. So looking at this cladogram here, which is basically just a diagram depicting different evolutionary relationships among different organisms based on distinguishing characteristics. So all of our organisms are listed along the tops of our branches here, and all of our distinguishing characteristics being used to categorize them are listed in this direction along the bottom of our cladogram here. So as we move through our cladogram, so from our first characteristic, which is tissues, to our fourth characteristic, which is protostome versus deuterostome, our grouping of our organisms is going to get more and more specific and more exclusive. So just as an example, let's start at this first trait here, which is the presence of true tissues. So any organism that falls beyond this point is going to have that characteristic. So they're going to have true tissues. So that means that everything falling beyond that point, which is our sponges to our chordates, have that trait. And the only organism that doesn't is the group that falls before that listed trait, which is our tyrannoфlagellates. And just as reference, tyrannoфlagellates are unicellular organisms which form colonies. And so they don't have differentiated tissues. And because they don't have that trait, that means they're also excluded from all these other traits as well. So there's no form of symmetry, body cavity, and they're not considered protostome or deuterostome. On the other end of our cladogram is our chordates. You can see that because they are past all of our listed traits, they're going to have some variation of those traits. So our chordates are eumetazoan, which means they have true tissues. They are bilaterally symmetrical. They are eucoelomate, so they have true coeloms. And all of our chordates are deuterostomes. So let's review the difference between protostomes and deuterostomes, which is just another way that we characterize organisms based on differences at their embryonic developmental stages. And we see differences in their cleavage, their coelom formation, and their fate of their blastophore. So protostomes undergo spiral cleavage at the 8-cell stage, where that top layer of cells actually rotates. And because of that rotation, we actually have determinate cell formation, so we can track what those cells are going to differentiate into. So for example, we could say that cell 1 will go on to form gut lining tissue. That's not the case for our deuterostomes, however, which have radial and indeterminate cleavage. So that radial symmetry keeps the cells indeterminate or undifferentiated. So what that means is that deuterostome cells at the 8-cell stage can differentiate into any type of tissue. The coelom formation is also different between both groups. So in protostomes, we have the mesodermal mast tissue that forms the coelom. It splits to form the coelom. And in deuterostomes, we have our gut tissue, or our yellow layer, folding in on itself and pinching off to form the coelom. We also have differences in the fate of the blastophore. So the blastophore is that opening at the bottom of our cell there, and that either develops into the mouth or anus. So in protostomes, the mouth is developing from the blastophore, and in deuterostomes, the anus is forming from the blastophore. Good way to remember the difference between the two is by looking at the roots of the words. So proto means first, and you might remember from plants our stomata. Stoma means mouth. So that means our mouth is developing first. On the other hand, deutero means second. Stoma still means mouth. So the mouth is forming second and the anus is forming first. So taking a look at this cladogram specifically with deuterostomes listed, we can see our 3 different subphyla of chordates listed. So we have our more primitive deuterostomes, or more primitive chordates, I should say, our cephalochordata and our urochordata, which are lancelets and tunicates. And then we have our vertebrata, which is basically everything from our sharks down to mammals. So although these three subphyla are very morphologically different, they have very different lifestyles, they are all chordates, which means they all have those four shared characteristics at some point during development, which are the dorsal hollow nerve cord, the notochord, the postanal tail, and the pharyngeal gill slits. So this is just a zoomed in image of that lancelet, which is our primitive chordate, showing all of those four shared characteristics. So moving into our specific chordate that we'll be dissecting, the sound that hopefully you're hearing is the mating call of the male leopard frog, or Lithobates pipiens. There is a distribution range that I provided and some taxonomy information as well. This species tends to have larger females than males, and again, the males are the only ones which have the vocal sacs, which are used in mating and communication. So our frog is supported by a skeleton. There is an axial and an appendicular skeleton. The axial skeleton provides the linear framework. So that's going to be consisting of our hyoid apparatus, our skull, our sternum, and our vertebral column. Our appendicular skeleton consists of bones that make up our appendages. So those are our pectoral girdle and anterior limbs, and the pelvic girdle and posterior limbs. So all of the skeletal structure is supported by muscular structure. Muscles can either be attached to the bones themselves or to other muscles via a tendon or an aponeurosis, which is basically a connective sheet. And on each muscle is an origin and an insertion. So the origin is the fixed end of the muscle, and the insertion is going to be the moving end of the muscle. The next slide is going to show a video of how the skeletal structure and the muscular structure support one another. So we can see the leg muscles are providing all the power in the jump, but then our spine is going to be providing stability. And there's actually a special joint in our frogs that you'll see when you go to dissect called the iliosacral joint, which allows for extension of the spine and the really large jumps that frogs are known for. Okay, so let's move into reproduction and development. So breeding for frogs is occurring in shallow fresh water. Adults will congregate in these ponds or pools and basically lay a lot of eggs. Males will croak and make lots of sounds to compete for females, and the females can actually lay up to 5,000 eggs per clutch underwater. Fertilization is external, and the eggs will hatch within 5 days. So you can see all those egg masses present in the video and the picture below. And so looking at the development of those eggs after they hatch. So eggs will become tadpoles, which are primarily all aquatic, and then they'll eventually develop hind legs, and the tail will be resorbed back into the body before that tadpole develops into a young frog. So some of the benefits of metamorphosis is limiting interspecies competition. So our adult frogs have a completely separate lifestyle and diet from our tadpoles. So our adult frogs are opportunistic feeders, like we'll see, and our tadpoles are primarily eating decaying plants and things in the water. Continuing with our reproduction, frogs are gonochoristic, which means they're separate male and female sexes. Some species are sequential hermaphrodites, however, but because it is sequential, they still are gonochoristic, so they're still separately male or separately female at a given time. So there's two forms of sequential hermaphrodism. One is protandrous and one is protogynous. And a good way to remember those terms is, again, by looking at the roots of the words. So proto, again, means first, and androus means male. So protandrous species begin their life as male and then develop into a female later on in life. There's some benefits of that. Larger females produce more eggs, which are more costly to produce. On the other hand, we have protogynous species, which are female first and male second. And that's going to occur in a lot of species that have competition for reproduction. So you want a larger male to compete. And we're seeing protogynous reproduction or sequential hermaphrodism in the common frog, which sometimes develops into a male after mating successfully as a female. So moving forward to respiration, Frogs can undergo various forms of respiration. The primary form that they use is cutaneous respiration at the skin surface. So the vessels we can see in that picture to the right are really close to the epidermis, and that allows for effective and efficient gas exchange at the skin surface. The next form is bucopharyngeal respiration, where basically air is brought in through the nostrils and kept within the buccal cavity, which is also highly membranous and allows for gas exchange within the mouth. The third form is pulmonary respiration, which occurs in lungs. And because frog lungs are poorly developed, it's a little bit more complex. So I'm going to describe it in more detail on the next slide. So the video at the top is showing how a diaphragm works. So it contracts and fills the lungs with air, but frogs lack diaphragms, so they have to have a different mechanism for bringing air into the lungs. So air is brought into the mouth and the nostrils are closed. Air is then forced down the throat, down the glottis into the lungs by pushing up on the buccal cavity. So contracting the buccal cavity and forcing the air into the lungs. And then once that buccal cavity expands again, the lungs will contract and force the air out. So that's our third type of respiration in frogs, and that's known as pulmonary respiration. We have different circulatory schemes in various vertebrate species. So when we dissected sharks, we had a two-chambered heart, which has a single circuit of blood flow. This is different in our amphibians, which have a three-chambered heart. So there's pulmonary and systemic flow, pulmonary flow being flow from the lungs back to the heart, and systemic flow being flow to the rest of the body and back to the heart again. There is some mixing of deoxygenated and oxygenated blood in our amphibians. But if we consider that there's multiple ways for them to acquire oxygen and respire, that might help to explain that. And lastly, our mammals have four-chambered hearts. Again, they have pulmonary and systemic flow, but they have complete separation of deoxygenated or oxygen-poor blood and oxygen-rich blood. Okay, so moving forward to feeding and digestion. So there will be a video that's going to show how frogs catch their prey. Frogs are opportunistic feeders, meaning they basically eat anything that they can catch, including insects, slugs, snails, even other frogs, birds, rats, and snakes. So there's images down below of the video of a frog that's eaten a snake and a bullfrog that's eaten a rat. They do stalk their prey by waiting at the edge of the water and then snatch them with their sticky tongue. Again, tadpoles are eating a different diet of algae and rotting plants primarily. And then you can look at some of the anatomy of the mouth to see how they are able to catch and grab onto their prey as well. So you'll notice that the tongue is actually attached to the front of the mouth and will flick outwards. We also have these maxillary and cormorant teeth on our upper jaw. And because frogs swallow their prey whole, these teeth are really just used to grasp their prey. At the back of the throat, we have the gullet or esophagus and the glottis down at the bottom, which aids in that pulmonary respiration. So air is forced down the glottis when it opens. We also see the eustachian tubes, which are the inner openings of the ears that you will see externally. These are parts of the digestive system, including the gallbladder, stomach, pancreas, liver, small intestine, large intestine, and spleen. So these all play a role in the digestion of the food that the frog consumes. And then some of the external features that you're going to want to notice are the nictitating membrane on the eyeball, the tympanum, which is the external ears leading to the eustachian tubes internally, and the nares or nostrils, which are external as well. And then here is an image of a fully dissected frog, just to help guide your work when you go ahead and do this yourselves. A lot of our frogs that we got are pretty small, so we might not be seeing these fat bodies, which are making up the bulk of the internal anatomy on this picture here. That's it from me. Thanks for listening, and good luck with your dissections.